We begin this lecture by taking a look first at the human cell. Chapter 1 in your textbook has a review of this content. There's also this link that I have here at the bottom of the page, and it's also linked on the Moodle page. It's important that you understand all the functions of the various organelles and have a kind of sense of the organization and function of the cell. So we'll spend a lot of time looking at the role of the mitochondria, the nucleus, ribosomes, especially the ribosomes not just free but attached to the rough endoplasmic reticulum. So in this lecture here, I'm not actually going over them. This is something that is expected that in your A&P1 and again to some extent in your A&P2 you would have reviewed the function and of both the cell as a whole and the function of all of these various organelles. So if you're weak in this area, Chapter 1 has this material in there, and again I encourage you to use the resources and go back. We begin here with this so that you understand that all disease process, as far as we're concerned in this course, begins at the cellular level. Arguably, disease really begins at the genetics level, at the gene, at the chemical level, and we might even talk a little physics if we really wanted to talk about the processes of disease. But for our intensive purposes, we're going to really begin at the cellular level. We do have a genetics section that we'll cover, so we will talk a little bit about the genetics of disease, but primarily you'll see that you're going to hear a lot more about the cell before we ever get into the actual systems and disease processes. So we begin the course feeling like you're back in A&P again, talking about cellular things. And from there we're going to move from the cells. We look into how things move across the cell, cellular transport, understanding these mechanisms of how fluids move in and out of the cell, how exchanges are made, how cells communicate. So cellular transport's important. That's also in Chapter 1. Also review content. So there's several different links that you can take a look at here that can be helpful to you as well. If you've forgotten some of these transport mechanisms, what's the difference between active transport in which ATP is used versus passive transport in which we're looking at either movement along a gradient, concentration gradient, or an electrochemical gradient. So be sure you have a basic understanding of these. I will tell you they're not technically testable material. I mean, your first quiz might have a couple things about organelles, but in general, and certainly by your first exam, I'm not asking you about what the Golgi apparatus does. Now, if subsequent lectures deal with specific cellular organelles, as you'll see when we do metabolism and look at the mitochondria, well then you'll be responsible for the information on the mitochondria. Then I'm not really going to Chapter 1 material, but rather I'm dealing with what we're talking about at that particular point in time. So I don't really go to Chapter 1 for exam material. What we're going to cover here in a moment with metabolism and the subsequent lectures, that's really where you begin the course content. So this is just a little bit of review reminders. Again, take a look at Chapter 1, take a look at your old A&P book, and you should be able to get up to speed on all of these. And again, of course, you need to know your body tissues. We're going to be moving forward through all of these. So this is, again, just meant in Chapter 1, but not really something I'm testing on specifically. Where we really begin this course is with cellular metabolism. And the reason we're here, so we recognize that disease begins at the cellular level. And so that's why you need that content. But then you're going to come to find that so many disease processes interfere with the metabolism of our cells. And so what we're really looking at is aerobic respiration, which you may have learned about in A&P1 and touched on a little bit maybe in your A&P2 and your cardiovascular and respiratory chapters. But it's really important that you understand this process. So we'll have subsequent lectures that go through glycolysis, and then we usually put the conversion of pyruvic acid in the citric acid cycle, or the Krebs cycle, as it's sometimes called. Those go together in a single lecture, and then one on the electron transport chain. So what I'm doing right now is just giving you a little introduction to cellular metabolism, some background information, some content you'll need to know, again, kind of moving forward. So understand that when we look at problems with thyroid, it's going to be our first real disease area is when we're looking at hyper and hypothyroidism. Those are diseases that interfere with normal cellular metabolism, and that's the manifestations that we see. So when a person has a disease going on, we are digging way back deep. We're not just saying, oh, well, when you have heart disease, your heart doesn't function. Well, we're going to be looking at cardiac muscle cells. What's happening metabolically in cardiac muscle cells that heart no longer functions correctly. When we look at fluids and electrolytes, we'll look at those, how fluids and electrolytes interfere with the normal metabolic processes of the cell. So that's why we begin here. If you've had micro already, then some of this will be very familiar to you. The metabolism that occurs in bacteria, microbes, is not all that different from what occurs in human cells. In fact, what we learned about how human cells function, we first learned from bacterial cells. So just a little overview, some terminology that you need to be familiar with. We are going to deal with, again, that term metabolism, basically all the different chemical reactions that occur in the body. And we can move that into two categories: catabolism. And catabolism, or catabolic reactions, occur when molecules break apart. And so we also can sometimes refer to them as exergonic. That means that an exergonic reaction, or a catabolic reaction, releases more energy than it consumes. So energy is released from that. So what we're actually about to start talking about, glycolysis, Krebs, and ETS, those are all catabolic reactions. Reactions in which glycolysis, lysis, we're really talking about taking a glucose molecule and splitting it apart. And then once we've split it first in glycolysis, we're going to split it further. It's no longer glucose anymore, but it's the subparticles of glucose, and we'll split it further and gather more energy from it. That's how we get our energy when we break something apart. In fact, it's not specifically listed here, but whenever we use ATP and we break off a phosphate of ATP, as you'll see in a moment, it's an exergonic reaction. The reason we use up ATP is to get energy from it. So when we break apart bonds, we get energy. So in glycolysis and the subsequent reactions of anaerobic respiration, what we're basically doing is saying we're taking the food we bring in, glucose and the carb or whatever, wherever it comes from. We generally try and get all of our nutrients. If we eat proteins and fats, the body tries to convert everything to a glucose or a simple sugar. It's the most efficient way to get energy. Now, there are pathways to get energy directly from proteins and directly from fats. We're focusing just on one pathway in this course, glycolysis, but it is the most readily and commonly used pathway in the body. So we've got these catabolic reactions. We take glucose and we split it apart. The thing is, the energy that's released from that glucose is not in a form that we can use. So yeah, I need a candy bar, it'll give me energy. So I eat my Snickers, and then what happens is the glucose that's in there is providing energy by taking those bonds, breaking them, and then capturing that energy and putting it in a form in which we can use. So that's what we're going to be seeing going forward. So the energy we get allows us to move, allows us to grow, allows muscles to contract, our sodium-potassium pump that keeps our cells in homeostasis, needs it, all movement into and out of cells. Generally, I shouldn't say all, most endocytosis and exocytosis processes require ATP. Very rarely do we say all anywhere in the body. Then what we have on the other half of the catabolic reactions are the anabolic reactions, where we combine them together. And so that's really what we're doing when we make ATP, so we can use ATP. So we would call those endergonic reactions, where they consume more energy than they produce. So the difference here is we can now take and bring energy into the bond. So what you've got is you have the glucose that's split. It released energy. We then took that energy in an anabolic reaction and formed ATP. So we brought it in, consumed it up into the bond, and now it's stored. So we went from what will have been, and you'll see here in a second, we go from ADP to ATP. So that's the constant ongoing reactions. We split it apart, then we put it back together, split it apart, and put it back together. And in this process, we use energy and store energy. And so a lot of times you'll hear expressions like, well, I need something to charge up my batteries. I need some energy to charge my batteries. Well, that energy that we're talking about when we eat literally does that. Our battery is actually in the form of an ATP. And so that ATP molecule is our charged battery. And when we deplete it, it becomes ADP, and it may even become AP, which you'll see in a second. So here's our battery. I call it the energy currency of the cell. That's ATP. This is what we can use. So glucose is not something our cells directly use. It's something we can capture energy from and then take that energy, convert it into ATP, and now we have a really quick, ready to capture energy source. And that's what ATP is. So really complex molecule. Do not worry. You do not have to recreate this molecule. You do not have to recognize it. For chemistry lovers out there, I like to throw in a few chemical pictures every once in a while. And so especially if I have some biomedical science students or nuclear medicine, sometimes the radiation therapy, take the chemistry course. And so just like to entertain a little chemistry once in a while so that you never forget that when we're talking about cells and we're talking about molecules, that everything can be boiled down to chemical equations that look like this. So ATP is what we're looking at, adenosine triphosphate. So here's a little bit of a summary of how these bonds form. The PO4s are your phosphates. I don't have the actual charges listed on there, but PO4 is the charge. So the adenosine, the adenine and a ribose, that's the basic backbone of our molecule. And so what you have attached to that then are 3 phosphate groups, which make the triphosphate. So the 2 weaker bonds will equally split off. They're pretty weak, so you can give up one, break that bond, the phosphate goes on off into the cytoplasm, into this the area, the matrix of the cell, depending upon where we're at, and can be rejoined up again. It's a reusable molecule. It doesn't automatically necessarily break apart. It can, but it often just releases and then binds back up. Release and, back and forth kind of a sensation. So it's easy to go to diphosphate or even to the single phosphate. This last bond is a very strong bond and is very rarely broken. So the conversion from ATP to ADP is extremely crucial, as I say. It releases a whole lot of kilocalories or calories per mole. Again, a chemical number. I'm not asking you to know that. I'm just trying to put it into perspective. So a single bond energy is about the same amount of energy as in a single peanut. Now you may say, well, big deal. How much is a peanut? But then let's talk about how incredibly small a thing we're talking about and the trillions and trillions of ATPs that exist in your body. Within a single cell, there can be thousands and thousands and thousands of ATP. Each one has as much energy as a single peanut. Then it seems like it's a little bit more. So the reactions that we're going to be looking at as we move forward, looking at glycolysis, Krebs, or the citric acid cycle, and the electron transport system, or sometimes called phosphorylated oxidation. Those reactions that we'll have 3 separate lectures on, oxidation reduction reactions are a big component of that. And so here are your definitions. This is important that you know this. Oxidation is always paired with reduction. They're often called redox reactions. So oxidation is the removal of electrons, and in a chemical sense, it's electrons. But in a biophysical sense, in the human sense, we're really dealing with hydrogen atoms. So instead of saying removal of electrons, we usually just say a removal of a hydrogen. So anytime there's an oxidation, it means that something broke free. So energy is released, it's exergonic, but that also means that that molecule that once had a bond attached to it, say it's glucose or whatever, it had a bond attached to it. As soon as the bond breaks, energy is released, but now the potential energy of what's left is less. So that's what this decrease in potential energy means. As soon as I have something and I give up one part of it, I give up a hydrogen, well, great, I release some energy, but now there's less energy left in what's behind. So in the kind of reactions that we're going to be looking at, when the hydrogen atom is given up, it's transferred by coenzymes. And we have two major coenzymes, and I do not even need you to have these names memorized other than to know NAD and FAD. So we're going to be looking at those. So in other words, when hydrogens are given up, they're going to be carried or transferred by these coenzymes. So what we'll do is we're going to oxidize glucose, release some energy from it. So what happens to the hydrogen atoms? Then some other molecule, in this case the NAD and FAD, will be reduced. And this will start to make more sense once we start looking at these reactions. Basic understanding: oxidation, we lose a hydrogen; reduction, we gain it. And so sometimes that seems funny, to reduce something like a reduction reaction seems like it should be just the opposite. We shouldn't be gaining something, we should be losing something because we're reduced. So that can confuse you. So just get it, kind of say it over about 10 times in your head. Oxidation, giving it up, oxidizing, to release or remove a hydrogen. Reduction is to gain the hydrogen, add it to it. And again, we're going to increase energy there. A reduction reaction increases energy. And as I said, what we're really going to be seeing a lot of is the NAD will pick up the extra hydrogens and become NADH. Okay? And so we always tend to write it as NADH plus H. There's usually an extra hydrogen. It's usually not one but two that's picked up, as you'll see here in our reaction, and one of them stays attached and one of them becomes free. So in the reaction of lactic acid to pyruvic acid, or back and forth. So this is a little bit strange, and when we go through glycolysis, our end product is pyruvic acid. If oxygen is not available, it'll become lactic acid. And hopefully you've heard that before. That's not the first time you realize with glycolysis that if there's no oxygen present, the pyruvic acid will convert to lactic acid. Lactic acid can convert back to pyruvic acid, however, so we can go back and forth. And in that process, what you'll actually have, if lactic acid is oxidized to form pyruvic acid, then you have an increase in that hydrogen energy being picked up. This is an important concept because, remember I said, glucose, our molecule coming from our candy bar, or our hamburger bun, or our piece of toast, or whatever it is that you might see as a simple carb, that glucose contains energy that we need to take to form ATP. But it's not simple, it's complex in process. So we take the energy when we split the glucose, we carry the energy around until we can convert it to ATP rather, and NAD is going to help us do that. So this is an example only of an oxidation reduction reaction. And the final thing we'll talk about here in this lecture is the enzyme categories, because we'll need these to move forward in looking at each of these processes. So very quickly, enzyme can be broken into categories, and I'm not asking you to learn the names of any specific enzymes, but we're going to categorize them as we go through the process. We have transferases, which transfer substances from one molecule to another, and we're going to specifically look as kinases. You've heard it as kinases or kinases. Either way to say it is correct. In a kinase, you actually transfer a phosphate specifically. So we'll see that process. Hydrolases release water molecules. Lyases, I'm sorry, I have to step back. I said that backwards. Hydrolases can either release or add a water molecule, so they're not just release, they're adding. Hydrolases is an older term, and a lot of times the next two replace that category. I've kept it in here because in previous classes I've used that term hydrolase. You may have seen it in your micro class, but it can either add or remove water molecules. But it's an older category, older chemical category, more or less replaced now. Lyases break molecules in half or apart, not necessarily half, but break them apart. Ligases bring molecules together. Isomerases rearrange molecules. And oxidoreductases are enzymes in the category that allow for oxidation reduction reactions to occur. And oxidoreductase enzymes are very powerful enzymes. So enzymes, remember, they speed up, slow down reactions. Hopefully you already knew that. And these are the categories you need to be familiar with. So rest assured, your first quiz will ask you about categories of enzymes, what their function is. So remember, transferases transfer molecules from one part to another. Kinases specifically transfer a phosphate from one part of a molecule or one substance to another. Hydrolysis, water is either added or removed, an older category, but we're still going to use it. Lyases break large substances into two or more smaller parts. Ligases bring them together. Isomerases rearrange, and oxidoreductases are necessary for oxidation reduction reactions. Coenzymes, we say, they define the behavior of the enzyme. In this case, they're small organic molecules. Many come from vitamins. So that's important to know. The FMN, the NAD, FAD are all linked to different vitamins. They are essential in helping control the activity of an enzyme. And in our case, we call them electron carriers. And so, remember, I might skip back to this one, it's not just electrons, we really mean hydrogen atoms. So these are going to carry energy for us. They're going to, in many cases, you're going to see them being either reduced and then maybe later on oxidized to release their molecules, but they're going to carry hydrogen atoms for us to help transfer that energy to help us make ATP. So this is the processes of the next lectures. We'll go through glycolysis, then we'll do 2 and 3 kind of together, formation of acetyl-CoA and the Krebs or citric acid cycle, and then the electron transport system. So this is a picture from an A&P book, actually, and we'll go through that process and products and all the different products formed in each step. This is a good summary slide for that.